I go ahead. Everybody, welcome to the Mangiagalli Journal Club. It's not only a pleasure, but essentially is a great honor to introduce to you Professor Koren, the today's speaker, that represents with his findings and milestones in the research on the role of microbiome in reproductive life. Henri Koren received his PhD from Tel-Aviv University, where he was trained in the field of microbial ecology at the level of Professor Eugene Rosenberg. At that time, it was part of an innovative paper, the Coral Probiotic Hypothesis, which has now expanded to the hologenome theory of evolution. Then moved to Cornell University as postdoc in the lab of Dr. Ruth Ley. During his postdoc, he was part of a NIH Human Microbiome project and also led the first study to demonstrate that pregnancy is associated with a deep alteration of the gut microbiota and osteometabolism. Recently, Professor Koren went back to Israel and joined the Faculty of Medicine at Bar-Ilan University and received a prestigious career integration grant, Marie Curie. Now, Professor Koren continues to study the human microbiome and specifically the female microbiome. His lab is very interested in the interplay between the endocrine system and the microbiota. In this paper, we can see a certain kind of prophecy where the potential relationship between the microbiome and the great obstetrical syndrome was taken in consideration. Immediately after first kind of announcement, it was published by Cell in this paper by Professor Koren. That is in absolutely clear manner demonstrated a change in maternal microbiome with relevant metabolic modification during pregnancy. 5 years later, always on Cell, Professor Koren was able to identify what is responsible in microbiome for metabolic changes during pregnancy. It will be for us really exciting to listen directly from Professor Koren the entire process of these scientific discoveries. And for preliminary question, we would ask what is first, the metabolic change or the microbiome change? And as a second main question, how long is the effect of the microbiomic change on maternal metabolism? Is it only transitory or it is permanent? In this paper, Professor Koren, we can see the microbiological change during pregnancy, birth, infancy, especially focused on the first 1,000 of life of a human being. And the main hypothesis have been made on microbiome influence, for instance, on preterm delivery and on the hypertensive disorder of pregnancy. And it is easy to understand, to imagine what microbes do when they can ascend from the vagina. It is more difficult to understand how gut or oral microbes can be translocated to the placenta. An important paper by Kirsten Haggard, published on Science, claimed the placenta harbors a unique microbiome. But this issue was not confirmed by other authors, as recently published on Nature, with a big clamor in the scientific world, as you can see it on the editorial comment of Nature on the previous paper, raising many doubts on the mechanism that produce the neonatal microbiome. In this paper on the British Journal of Obstetrics and Gynecology, a deep microbial analysis of multiple placenta was performed, and the question on placental microbiome was raised again. Professor Corren and his group in this paper showed that the sterile womb hypothesis of the beginning of the past century is even or actual and holds even today. Now we are ready to listen to the lecture of Professor Corren.
Okay, thank you very much for the invitation. I think my friendship with Enrico Ferrazzi started about 6 years ago, and since then we've both organized a meeting together, and I gave a talk in Milan, and I wish I could have been in Milan now, but we'll wait for COVID to pass. Do you see my screen?
Yes.
Yes, we can see it.
Good. Okay. So I will take the next 45 minutes to 1 hour to basically tell you about my journey in the pregnancy microbiome and how I got to this field of study, and tell you what we've done and what we're doing right now. So we'll go a bit into the field of the microbiome, then the microbiome in pregnancy, what's known already. I'll then describe a few studies that we are getting ready to publish now on the on gestational diabetes. And then I will also show you some interesting findings we have about infants. So I started out in, let me see where my mouse is. Okay, I started out looking at the gut microbiome because other people were very deeply involved in the vaginal microbiome. But today my lab basically studies all microbiomes. So from each woman we get a gut sample, we get a vaginal sample, and we get a saliva sample. So we're trying to get a more holistic picture. And this is mainly due to the reduction in costs and the automation and high throughput technologies that we have now, which allow us to sample many more samples and work through them quickly. Now, in this presentation, before I start, I just want to say 3 things. Pregnancy is not a disease, although I will show you that from a bacterial point of view, the microbiome in pregnancy behaves just like a disease microbiome. Please remember that antibiotics save lives, although I'll show you what they do to the good microbes, but we would not be here without antibiotics. So please remember, antibiotics save lives, and I have nothing bad to say about antibiotics. And I'll also try to convince you that the microbiome is the only important thing, but it's not, okay? We are complex individuals. We have other systems within our body, but it's very easy for me, for the sake of this talk, to convince you that the microbiome is basically responsible for everything. So that's not the real case. Now, microbiome field. The microbiome research field started exploding around 2005. And it happens for 2 main reasons. The first reason was a change in the way we think. Okay, we were all taught, and we still teach in some med schools, that bacteria are bad for you. Okay, bacteria are bad guys, but this way of thinking is changing, right? Bacteria, there are no good and bad bacteria anymore. Bacteria do not act alone. Bacteria are part of communities, and these communities are in touch with us and react to us, and we react to them, and they react to each other, and it's an entire ecological system. Now, if this was the bad guy, you can see here that it's really part of a community. For example, if we take Helicobacter pylori, Helicobacter pylori used to be taken as a bad guy, but we now know that most of us have it in our stomach microbiome. And unless the situation changes and we're stressed or we don't eat good, nothing will happen, okay? It's part of our normal microbiota. But if we are stressed for a long time, then this bacterium may sense that and start activating virulence genes, which eventually would lead to a disease. So we're going to talk about a little bit about this during the talk, how a bacterium can sense a change in a situation. And one probably last technical slide is, as most microbiome people are ecologists. And as ecologists, we look at different taxonomical levels, so we don't always look at the species level. We don't always look for an E. coli here or an E. coli there, but a lot of times we look at ratios between communities. For example, 90% of our gut bacterial community is made up of firmicutes and bacteroidetes, and the ratio between these two phyla, each of which has hundreds to thousands of different bacteria, has importance in disease. Another interesting phylum is Protobacteria, E. coli, Shigella, Salmonella, Listeria, Vibrio. All of these guys with bad names all belong to the Protobacteria, and this is one of the reasons that in disease states we see an increase in Protobacteria. So you've probably all heard the different cliches that we are more microbial than human, that these bacteria carry 150 more times genetic knowledge than our own. The majority of them are inside our intestine, and on a day-to-day basis, there's a symbiosis. These guys provide enhanced nutrition, meaning they degrade nutrients we don't know how to degrade. They protect against pathogens, both by educating the immune system during pregnancy and infancy, and both by habitating certain niches along the way, which do not allow other bacteria from the outside to colonize, like in the case, for example, of Clostridium difficile. These bacteria also create vitamins, create hormones, and this is something, as mentioned before, we are very interested in. So can bacteria produce hormones that we can sense, and can bacteria sense hormones that we produce? And we'll touch upon that soon. So there's been a big change in the way we think. We don't think of bacteria as just bad anymore and just disease-causing agents, but they're an integral part of our life. Another thing that happened in these last years is that we moved from culture-dependent techniques to culture-independent techniques. What do I mean? On a petri dish, as you can see on the screen, you can culture less than 1% of the entire bacterial community. Okay, so 99% of the bacteria in the world are uncultured. This means that we have to develop certain techniques, molecular techniques, which allow us to figure out, without culturing any bacteria, which bacteria are there. We can know what these bacteria are doing, and we can now even know which metabolites these bacteria are producing. So without growing a single bacterium on a petri dish, we can describe entire microbiomes containing trillions of bacteria down to the product that they make and to the interactions that happen between them. And this is all due to technology that was developed mainly for the Human Genome Project, But when it ended, the NIH decided to embark on a new project, which is called the Human Microbiome Project. And again, as mentioned before, in my postdoc, I was part of this project, and we sampled 250 healthy Americans. Males were sampled at 15 body sites, and females were sampled at 18 body sites. And we basically sequenced their entire microbiomes to see what was going on and get a real picture of a healthy American human adult. 5 years of work, and this is the basic, the main results. What do you see here? This is a PCOA, and every circle on this PCOA is a microbiome. Oops. Every circle is a microbiome, okay? And the closer these microbiomes are, the more similar the microbiomes are. The further these dots are, the more different the microbiomes are. And then we can see, for example, here that all of the gut microbiomes from 250 individuals clustered together, and the oral microbiomes clustered together, and the vaginal microbiomes clustered together. And the skin is a bit more problematic because we now know that the skin has different conditions around the body. But we can basically see that every body site has its own unique microbiome that evolved with it to conduct certain functions. It's even more than that. It's not just that the oral cavity is different than the gut. If we look within the oral cavity, where we can sample different sites, we can see that the tongue microbiome is different than the cheek microbiome, which is different than the plaque microbiome and different than the saliva microbiome. So millimeters apart, we've got different microbiomes that have adjusted to perform certain roles. Now, this is in the case of symbiosis. But today we have a growing list of dysbiosis states. Dysbiosis means a shift in the microbiome. Dysbiosis does not mean, and I want to emphasize this, dysbiosis is not a cause and effect, okay? If a person with Crohn's and colitis has more proteobacteria and more actinobacteria, this does not mean that proteobacteria and actinobacteria cause IBD, okay? But this is something that is correlated to the disease. Clostridium difficile infection, we see an increase in proteobacteria. Necrotizing enterocolitis, increase in proteobacteria. Metabolic syndrome, increase in proteobacteria. And this list is growing longer and longer every day. Not always do we see mechanisms, but the list of surveys that show that this patient has this microbiome, this healthy person has a different microbiome, this list is growing. But probably the most studied dysbiosis state is obesity. And what do we know about obesity? We know that individuals with obesity have a higher firmicutes to bacteroidetes ratio. We said before that 90% of our gut microbiome belongs to either firmicutes or bacteroidetes. Well, obese individuals have more firmicutes and less bacteroidetes. And again, this is just a survey, but what does this mean? So for us to understand what this means, we can take germ-free animals, which are animals that we grow in sterile conditions and have never seen a bacterium in their life, and we can transplant the bacteria from these humans to these mice and see the phenotype that occurs. And what you usually see in this experiment is that the mice receiving the obese microbiome become obese, whereas the mice receiving the lean microbiome stay lean. And again, this is the most studied case of dysbiosis. This was also shown in a paper in Science in 2013 by the group of Jeff Gordon, which looked at twins. In each pair of twins, there was one obese twin and one lean twin, and they had different microbiomes. And then Jeff Gordon's group transplanted this into germ-free animals and saw the same thing. A mouse receiving the obese microbiome gained weight, whereas a mouse receiving the lean microbiome stayed lean. Now, the million-dollar question is probably, can we do the opposite? Can we take bacteria from a lean person and give it to an obese person and make him lean? The answer is no, not yet at least. But what we have shown is that we can prevent weight gain. So we did a big study with Twins UK, where we wanted to see the genetic effect of the host on its microbiome. We didn't find much about the genetic influence, but we did see that most lean twins had more of this bacterium, Crescente minuta. So we did the next experiment. We took feces from an obese twin and gave it to a mouse, and then we took the same feces, added Crescente minuta, and gave it to different mice. And we saw that the mice that received the feces that had Crescente minuta added to it did not gain weight, whereas the mice that received the feces from the obese twin with no Crescente minuta did gain weight. So we were actually able to prevent weight gain, and we are now trying to understand how this bacterium is doing this and what's going on within the microbiome that is allowing this. And since the past years, a lot has happened. We've identified many diseases associated with dysbiosis. We know that diet has a huge effect on the microbiome. We know that babies and elderly have very unique microbiomes, whereas adults have a pretty stable microbiome. But we are all here today to learn more about what's missing from this figure, and this is the pregnant woman. Now, you all probably know a lot better than me what happens in pregnancy, which means we know that during pregnancy, the female body undergoes hormonal, metabolic, immunological changes. We see an increase in fat early on in pregnancy, which is followed by reduced insulin sensitivity later in gestation. We know that pregnancy is considered to influence every organ in the human body. And in that case, what about the forgotten organ, the microbiome? And this is where I started to get interested in the pregnancy microbiome, because everyone at that time was actually studying the metabolic syndrome microbiome and obesity microbiome. And I showed you that bacteria play a role in part of the phenotype of obesity and metabolic syndrome. Now, if you look at pregnancy and metabolic syndrome, you can see some similarities. Both cases, we have weight gain. In both cases, we have insulin resistance. And in both cases, we have low-grade inflammation. So I decided to team up with a group in Finland, and we got samples from 91 pregnant women, sampled in their first trimester, third trimester, 1 month postpartum, and we had samples from their infants at 1 month of age, 6 months of age, and 4 years of age. We knew everything about these women, the dietary data, the anthropometric measures, what happens in pregnancy, what happens in previous pregnancies. And I analyzed this for a couple of years. And this was, again, one of the main results. Now, what do you see here? On the right, you see what we call alpha diversity. Alpha diversity is an ecological measure of richness. So we see that in first trimester, pregnant women have a more diverse and richer microbiome than third trimester. Okay, so as pregnancy progresses, we see a decrease in diversity. This, for example, is a characteristic of a dysbiosed microbiome. So in many diseases, in most diseases, we see a decrease in bacterial diversity. If we look at beta diversity, which is an ecological measure of similarity, and once again, the closer circles are to one another, the more similar the microbiomes are, we see that at first trimester, these microbiomes are very similar to one another. But at third trimester, 1 month postpartum, these microbiomes are pretty spread apart. And then you ask yourself, what's normal? So, do all women in the beginning of pregnancy look one like the other, microbiome-wise? And then as pregnancy progresses, we see a shift in the microbiomes? Or because there's such a big shift, hormonal shift in the beginning of pregnancy, then all of the microbiomes all of a sudden look one like the other. And then as pregnancy progresses, they go back to look like the original self. So I did a meta-analysis and compared the microbiome of pregnant women to the human microbiome project individuals. And we can see that the clustering pattern of first trimester is very similar to the clustering pattern of healthy individuals. So women go into pregnancy with similar microbiomes, but then during pregnancy, something happens and the microbiomes drift apart. If we look at which phyla change during pregnancy, that we can see that in third trimester, there's a huge increase and a significant increase of Protobacteria and Actinobacteria. Now, these two phyla might sound familiar to you because I described them previously in the case of IBD, of Crohn's and colitis. So this increase in beta diversity is a characteristic of disease. So diseased individuals will have a more different microbiome, one from the other, than healthy individuals. And Protobacteria, an increase in Protobacteria, we talked about it. It's in most cases a characteristic of a disease. But who is changing? Which bacteria are changing, not just at the phylum level? So we showed, using machine learning techniques, that in the first trimester, we have a lot of short-chain fatty acid producers. Short-chain fatty acids, for those of you that haven't heard of them, are the fermentation end products in the large intestine. So we're talking mainly about acetate, butyrate, and propionate. And this is what the bacteria, these are the end product of bacterial fermentation. And these short-chain fatty acids are considered very beneficial for the immune system, the neurological system, and for general health of a human. But as pregnancy progressed, these guys went down. And instead of seeing short-chain fatty acid producers, we started seeing an increase in opportunistic pathogens. Okay, Enterobacteriaceae, Clostridia, Enterococci, all these started going up. And we looked at the immune system, and again, this is not something that we invented. This has been known that the microbiome, the immune system is modulated during pregnancy. But these changes in the immune system, together with the increase in Protobacteria and opportunistic pathogens, and the increase in beta diversity, led us to ask what is actually going on here. What is the contribution of the microbiome to a healthy pregnancy? So I took samples from women in their first trimester and third trimester, gavaged that into germ-free animals, and mice that received third trimester microbiomes, gained more weight, developed insulin resistance, and had low-grade inflammation. So the third trimester microbiome, and we now know that it's the third trimester, it's not the second trimester microbiome, is partially in charge of some of the phenotypes that we see in pregnancy. Recently, we undertook a study together with a group in Finland, in Holland, in the Netherlands. And the aim of this study was to see what is the influence of the microbiome in pregnancies of women that have IBD. I won't go into that, but I will show another interesting result that came out of the study. And this is before I told you that as pregnancy progressed, diversity decreases, so the microbiome becomes less diverse. And we show here that it will never go back to its original self. So a woman in her first trimester, in her first pregnancy, has a more diverse microbiome than a woman in her second, third, and fourth pregnancies. Now, we don't know what these women are actually losing. We need to check for that, and how critical is what they are losing in diversity. But we do know that diversity changes with pregnancy. Okay, and then I decided to see, we've looked at Finnish women, Dutch women, what's happening with Israeli women. So we collected 400 women in their first trimester, second trimester, third trimester, and basically looked to see if there are changes in the microbiome. This cohort was actually built to look at the effects of gestational diabetes, and I'll talk about that later, but our analysis of a new sample set of these Israeli women in both their first and third trimester demonstrated significant differences in the third trimester microbiota as compared to the first trimester. We were able to observe significant enrichment in bacteria such as Neisseria, Blautia, Collinsella, and Bifidobacterium as pregnancy progressed. And Bifidobacterium is what you see here in the bottom right corner. And this is very interesting because Bifidobacterium, which increases towards the end of pregnancy, is the bug that knows how to degrade human milk oligosaccharides in the infant, meaning the infant, the offspring, needs to get this from its mother. And to study the mechanisms underlying the interaction between pregnancy and the microbiota, we next used a murine model. We looked at the pregnancy of female mouse. Pregnancy in mice is about 19 to 21 days. So we sampled them, as you can see, at 0, 4, 7, 10, 14, and 18 days. We also looked at the immune system. And the immune system within such a short period of time also changes in the pregnant mouse. And as expected, as pregnancy progressed in these mice, the pregnant female gained weight as compared to their control counterparts. And we saw dynamic changes in the beta diversity towards the day 18, towards the end of pregnancy. We saw several changes in taxonomic levels when comparing these days, but as we observed in women, the levels of Bifidobacterium significantly elevated towards the end of pregnancy. And this demonstrates that Bifidobacterium responds to delayed pregnancy conditions in mice as well as in women. Now, what's going on here? How does Bifidobacterium know that a woman is pregnant? So we decided to check the endocrine system and to explore the potential contribution of progesterone to the observed changes in the gut microbial compositions during pregnancy. We implanted progesterone subcutaneously, so in the back of their necks, as well as placebo pellets for 21 days. So these pellets secrete progesterone for 21 days, just like pregnancy, whereas the placebo pellets do not secrete progesterone. What we also saw was that germ-free animals, germ-free females, had greater levels of progesterone. And this has not been published yet. We're about to publish it. And we did not see this in males, okay? So the interaction between progesterone and the microbiome occurs only in female mice. Now, when we compared these mice that had the progesterone pellets and looked at their microbiome, we saw that they had an increase in Bifidobacterium. So Bifidobacterium not just reacts to pregnancy, but it reacts to progesterone within pregnancy, to the increased levels of progesterone within pregnancy. And I'll skip these. And what we did next was to check this in vitro, in a tube. We basically took fecal slurry from pregnant mouse and fecal slurry from a non-pregnant mouse and added progesterone and control. And we saw that once again, the fecal slurry that received progesterone had increased level of bifidobacteria, changes in alpha diversity and bacterial richness. So this means that progesterone is also responsible for the decrease in bacterial richness. Progesterone was also responsible for changes in beta diversity. But most importantly, we showed that progesterone increased level of bifidobacteria. And if you dig in the literature, you can basically see that this has been known. It's known that certain bacteria can take up estrogen, estradiol, and progesterone and start growing faster. And this is what we're doing in the lab now. We're doing growth curve of bifidobacteria together with progesterone to see if they grow faster. And indeed, some bifidobacterium strains grow faster in the presence of progesterone. And we're trying to understand what is happening at the gene expression level and what is bifido. How does it sense progesterone and why? What does it do with it? So until now, I talked to you about a healthy pregnancy, but I want to talk to you about something that we are studying now in 3 different aspects. One of them is the prediction of gestation diabetes using the microbiome. So can we already see changes in the microbiome in the first trimester before gestation diabetes is diagnosed? The second is we're following the saliva microbiome throughout pregnancies in healthy women and women with gestation diabetes. And the third is looking at the effect of diet on these women with gestation diabetes. So I won't spend any time on going on to what is gestation diabetes, but I will say that today we have no efficient way of predicting gestation diabetes for first trimester. So I'm taking you back to the cohort of Israeli women, these 400 women, out of which 40 women developed gestation diabetes at second trimester. But remember that we had samples from these women in their first trimester, right? So we collected fecal samples for microbiome analysis, saliva. We collected blood. We had surveys of diet and stress and exercise. We had their medical records. And besides just microbiome analysis, we did short-chain fatty acid analysis, which looked at the immune system. We looked at hormones. We looked at metabolomics, which metabolites are being secreted. And we did some association. We tried to predict gestation diabetes, and we wanted to check what is the influence of the microbiome on gestation diabetes. But let's start a bit with the diet. This will go really fast. We found significant differences between total carbohydrate dietary consumption in the control group at first trimester versus third trimester, as well as second versus third trimester. The control group had higher carbohydrate intake during the third trimester. Also, the consumption of carbohydrates in the control group of the third trimester was significantly higher compared to the GDM group at all 3 trimesters. And according to the 24-hour recall questionnaires, the control group ate less fat at first trimester than the GDM group at second and third trimester. And we also found that the GDM group significantly increased the fat consumption from the first trimester up to the third trimester. We also saw that women, gestation diabetes women at third trimester ate more protein than the control group in all 3 trimesters. And we found the GDM group at second trimester ate significantly more protein than women from control group at first and second trimesters. According to the data we collected from the diary questionnaires, the total energy consumption at the third trimester was higher then at the second trimester, and at the first trimester as well. And there was no significant differences between groups in any trimester for dietary fibers intake. But if we look at sugars, the amount of total sugar consumption, we found that control women ate higher amount at third trimester compared to the GDM women at second and third trimester, meaning this diet that these women were given was actually working. We are now really trying to analyze the metagenomic content of these microbiomes and which genes are going up and down in the microbiome according to the diet. But if we look, for example, at the diversity, there were no differences that were observed in the alpha diversity of the gut microbiota in each trimester. So GDM women do not have a less diverse microbiome than normal pregnancy in every trimester. If we look at beta diversity, once again, no differences were observed in beta diversity of the gut microbiota in each of the trimesters. So right now it looks like there's not a very big difference, and we're still digging into it. But we started out with digging into the first trimester because, as I mentioned, this is the first thing that is interesting to us. Can we see changes in the microbiome already in first trimester? The answer is yes. So in gestational diabetes women, we can see changes in short-chain fatty acids that we mentioned before already in the first trimester. Okay, you can see it here. You can see changes in acetate that are lower in the GDM women. We can see that valerate is lower in the GDM women. We can see that other short-chain fatty acids are also lower in the first trimester. So the microbiome, even before gestational diabetes is diagnosed, the microbiome behaves differently in women that are about to develop gestational diabetes versus women that will not develop gestational diabetes. And we saw the same thing from cytokines. So the inflammatory system, immune system is already starting to act up in first trimester before GDM is diagnosed. There are bacteria that change between women that will develop or will not develop gestational diabetes. And these three together already allow us to predict with 86% success rate, which together with clinical data, of course, which woman will develop gestational diabetes or not. And this is something that we are continuing to work on and trying to understand much better. But how is the microbiome contributing? And can the phenotype be transferred to germ-free mice using first trimester microbiome? So we took feces from women in their first trimester who will develop or who will not develop gestational diabetes and transplanted it to germ-free animals. Now, what we saw was that it resulted in changes in inflammatory markers. So we were able to transfer some of the inflammation already in first trimester by the microbiome, as you can see here, for example. And we were able to transfer the insulin resistance. Okay, so using the microbiome itself, in women that have not developed gestational diabetes yet, we were able to transfer characteristics of diabetes to mice. Now, a bit about the saliva microbiome, which is also not published yet. We looked at alpha diversity, again, bacterial richness, and as you can see that in the control group, we saw differences between the first trimester, third trimester microbiomes. In the diversity, we did not see any differences in the women with gestational diabetes. If we looked at beta diversity, we did see differences in both the control group and the GDM group between the trimesters in the oral microbiome. When we looked at who was changing using Maslin 2, we saw a significant increase in staphylococci and a significant decrease in Bacteroides, Clostridiales, Enterobacteriaceae, Lactobacillus spiraceae at the third trimester compared to the first trimester. So once again, the critical changes, as in the gut, in the saliva, too, happen at the third trimester. And we are now starting to complete this picture of the changes that occur. We know now that, as mentioned, the placental microbiome probably does not exist anymore. There was also a paper last week in Nature Microbiology, again, showing that it doesn't exist. I have many conversations with Kirsty Agard, who was the author on the original paper, and she still thinks there is a placental microbiome. So I guess this debate will go on for a bit longer. And for those of you who were actually at the Milan meeting 2 years ago, we had a debate between Kirsty and Matthew Horneff about the placental microbiome. But I did show you that pregnancy acts exactly as dysbiosis, right? Exactly as metabolic syndrome from a bacterial point of view. So it's a dysbiotic state, and the microbiome has a role in certain phenotypes. Bifidobacterium increases towards the end of pregnancy. Certain bacteria can sense hormones and react accordingly. The weight gain following progesterone supplementation is mediated by the gut microbiome. I didn't show that due to lack of time. And gestational diabetes is accompanied by changes in the microbiome, which can already be seen in first trimester. Now, I can stop here, but I really want to describe 2 more studies that talk about the kids, about the offspring, because these are the ones that get this dysbiosed microbiome. So we are inoculated at birth, right? Going through the birth canal, we get bombarded with bacteria, and these bacteria will accompany us through the beginning of life. And we know today that there are changes that are associated with babies that were either born via C-section or babies that were born vaginally. There's a big controversy on how long does this last. So if I was born via C-section, does this mean that for the rest of my days I'm doomed? And does this mean that this small change that we see right after birth, does this mean that I will be in greater chance to develop obesity, asthma, or allergies when I'm a teenager? We don't know. NIH is putting a lot of money to study that. In our lab, we don't see these changes around a month after birth. There was just a paper published in Cellulose Microbe by Frederick Backhead, who shows that at the age of 5, they don't see any changes between these children who were born via C-section or not. But this does not mean that there aren't other groups who think that this does matter. And this is a paper from Nature Medicine talking about vaginal seeding. And this paper basically says that babies that are born via C-section needs to have their microbiome restored to a vaginally, to be similar to a vaginally born baby. And if you Google it, this is the protocol. You basically sample the mom, you incubate the gauze in the vagina for 1 hour, you extract the gauze before the C-section, you then put it in the baby's mouth, wipe its face, wipe its body, and hope for the best. Now, the American College of Obstetrics and Gynecology had a note, a commentary out a couple of years ago that says, do not do this at home. There's not enough proof that this does more good than damage. I know that this group is now working on thousands of babies, whereas this study was looking at 4 babies for a month and a half. So we do agree, though, that babies are microbe magnets, and the first 2 to 3 years of life are really, really crucial for the development of a microbiome. And if you see on top here, the women from the Finland study, these are their children. You can see that 1 month of age and 6 months of age, the microbiomes are still pretty spread apart, whereas in 4 years of age, the microbiomes already cluster like the first trimester microbiome, like an adult-like microbiome. So I did a meta-analysis and I took feces from an infant that was followed for sampled for 3 years and compared that to the HMP healthy American adults. And what you can see here is that this point here is the meconium. This is the first stool a baby gives. And as the baby grows older, the microbiome becomes more and more similar to an adult-like microbiome. Besides a certain shift that we saw, which caused a change, a real big change in the microbiome and almost returned it to the beginning, but then it bounced right back. And this was when the baby got antibiotics. So what do antibiotics do if they're given in pregnancy or if they're given early in infancy? So we started out with a mouse experiment, and we basically, if we said that the mouse's pregnancy is 3 weeks, we started giving the mother penicillin for the last week of pregnancy and throughout lactation, which takes another 3 weeks. So the mother got penicillin for 4 weeks, and the babies, the offspring, drank milk from these mothers for 3 weeks, then were weaned them, and we sampled them when they were weaned and 3 weeks after in collaboration with a group in McMaster. If we look at the microbiome of the mothers, of course, they changed their microbiome because they got antibiotics, but it was very interesting to see that the offspring that did not get direct antibiotics, but only received milk from mothers who received antibiotics, had changes in the microbiome both when they were weaned and also 3 weeks after that, when they were around the teenage age in mice at 6 weeks of age. And then we did something that is very interesting to us. We looked at the gut-brain axis. Does the gut microbiome affect our behavior? We did 2 exams. One is called the open field, where you put a mouse in this box, and a scared mouse, an anxious mouse, will stay close to the walls, whereas a risk-taking mouse, exploratory mouse, will spend much more time in the middle. We also did an elevated plus maze, in which a scared mouse will stay in the closed arms, and a mouse that is risk-taking and not anxious will spend time in the open arms. Significantly, in both assays, mice that their mothers received antibiotics during pregnancy and lactation were less anxious and more risk-taking. And I want to just sum up with a study we published really recently. And this is about babies born that received antibiotics during the first 2 days of life. And we followed them. We got fecal samples from 1 month of age, 6 months of age, a year of age, and 2 years of age after they got the antibiotics. But our Finnish collaborators followed them for 6 years epidemiologically. And this is extremely interesting because what we saw, and we still are not sure how to explain it, is that boys, not girls, that received antibiotics during the first 2 days of life weighed less at the age of 6 than boys that did not receive antibiotics. We did not see this in girls, okay, only in boys. We saw this in another cohort, in the German cohort Peaches. We saw the same thing. If we look at the fecal microbiota during the first 2 years, we see that in the control group, you see sort of, by the way, this is not in revision anymore. This has been published in Nature Communication. I apologize for that. We see that the microbiome of the control babies is pretty organized, right? You see it starting from here, going into here, then here, then here. Whereas the antibiotic-treated babies have a completely chaotic microbiome. We saw differences in alpha diversity, bacterial richness. Of course, when a baby receives antibiotics, it has a less diverse microbiome. But then for some reason at 1 year and 2 years of age, there was a compensation for that, and they showed way richer bacterial microbiomes. When we looked at what was going on and what was actually allowing us to know at the age of 2 which baby got antibiotics 2 years prior to that, and I'm not saying that that is a bad microbiome and this is a good microbiome, I'm saying the microbiomes are different. The biggest changes we saw was a disappearance of a certain strain of bifidobacteria. Okay, we're now trying to isolate it to see if we can study what it means that it disappeared. Okay, so one, bifidobacteria disappeared during the first days of life and never came back. And we did the next experiment. We took feces from first trimester babies, babies who are 1 month old, who got antibiotics and who did not get antibiotics a month prior to that, gavaged it into germ-free animals. And even in this case, the mice that received the feces from babies that got antibiotics weighed less. And this only happened in male mice and not female mice. The same thing was true for 24 months. So babies that got antibiotics 2 years before caused mice to gain less weight than babies that did not get antibiotics. So this is transferable even to mice, and we're now really trying to understand what's going on here. But I really like this saying by Marty Blazier in Science that said that antibiotics can be likened to a four-edged sword against bacteria. So if until now we only thought about antibiotics as stopping the infection and stopping the spread to the community, and we knew about one bad edge of the sword, which is causing bacterial resistance, we now also know that it does harm the good microbiome. And I want to finish up by thanking this wonderful group of people who does all the hard work, my funders, my collaborators on the slides that I showed you. And if there's one thing you need to take from this talk is that humanity is a passing episode in the life of bacteria. They were here before us, they'll be here after us, and we need to take really, really good care of them. So thank you.
Thank you, Lord. Now, I think that there is room for questions, and I hope that your incredibly interesting presentation will make a lot of hands to rise among our audience. Well, I can open the discussion. Oh, Gabriele. Just with the last part of your presentation, you address how antibiotics in early life can change significantly the growth profile and other aspects of newborns and even later in life. So recently we read systematic review and meta-analysis on prophylactic antibiotics before cord clamping at cesarean section, and these guys were concluding that not just 1 but 2 antibiotics should be given 1 hour before cesarean section. And I think there is a dramatic underestimation of the impact of antibiotics on the life of newborns, just to save 1 in 100 infection of the cesarean sectional scar. So what's your opinion about this idea to give antibiotics to the fetus even before he's delivered?
Okay, so we still don't know about giving we haven't really looked at what happens in pregnancy. So we're now looking at both at women that got antibiotics during pregnancy and can we see that in the offspring later on? And we are also in mice looking at what happens if you give the antibiotics at different times of pregnancy. I still don't have enough data, but I do know that as I opened the talk, antibiotics save lives, and by no means am I a clinician, and by no means can I say whether it is useful or not useful to give antibiotics. I'm just saying that we need to take this into account, and probably, this is what we're working on, supplement with probiotics. Because, for example, in the study with the mice that I showed that the mothers received antibiotics, we were able to reverse the phenomena using probiotics. So I think that this is something that we'll need to work on much harder, giving tailor-made probiotics for each antibiotic that we give.
Thank you. Grazie.
Posso intervenire?
There was a question from Gabriele Rossi. Okay.
I'm really impressed by your talk, and I'm really grateful because you show us today a taste of your ongoing studies on gestational diabetes. And it's really, really important for us to note that, how to say, we are always looking at some blood glucose levels in the middle of pregnancy, and something very, very bad, wrong is going to happen at the beginning of pregnancy and starting with a cytokine process. So you introduce not only a new system to detect some metabolic change, but the point of view that we have to start looking at metabolism during pregnancy at the beginning with all the change. Thank you.
Thank you.
Is there any other question?
Okay.
One difficulty that we have as clinicians to approach the systematic comprehension of the metagenome and its impact on health and disease in pregnancy, let's limit it to pregnancy, is the abundance of names. So when we go down to the names of all the bacteria that are part of the populations who lives with us, we get lost. Okay, so really, every now and then one single name comes up and say, I am responsible for this, I can change this, I can change that. Now, at the beginning of your talk, you presented the balance between Firmicutes, Bacteroides, and Protobacteria. Might be that this is, the preliminary view of the balance of bacteria in the gut microbiome, at least. Okay, and the balance between Bacteroides, Firmicutes, and Bacteroides might be something we have to look at when we read papers, when we try to understand what's going on. What do you think? Because you live all day with these names and taxa, etc., and so you're familiar with them. But I suppose that even Agnese, who is interested in this, when he reads all the list of names, gets lost. So what do you suggest, just to begin to understand?
Okay, so even we get lost, because it's not just that. Different labs using different techniques can come up with different lists of names. So that even adds to the difficulty. And this is why today, so the method that you are describing is 16S. We basically sequence conserved regions in the bacteria gene. And then by the variable region, we know which bacterium it is. This is how we give the name. So today you see more and more studies pretty much moving away from 16S and going either to whole genome shotgun sequencing, which basically will give you all of the genes, or what we prefer is doing metabolomics. Because apparently, one pregnant woman will have Faecalibacterium, and another pregnant woman will have more of a Kermansia, but the metabolites are what actually matter. So it doesn't matter which exact names of bacteria there are, if the metabolites produced are the same or are not the same. So we're trying to move more towards doing metabolomics, especially untargeted metabolomics. And it's really hard because there's not much data in this field, and there are a few pioneering groups now trying to organize it. Most metabolites still don't have names, but I think that will help us much better because it will also allow you as a clinician afterwards not to go searching for a certain probiotic, but to give what we call a postbiotic, okay? To give the postbiotic, to give the product of the bacterium. So, you know, I mentioned short-chain fatty acids, which are metabolites. So why, if you see that there is a reduction in butyrate producers, why give a bacterium that produces butyrate? Why not just give butyrate? Right? So I think that is the way we are going. We're going into a world of postbiotics, and you will see many more studies starting to do metabolomics. The only problem with metabolomics is the high price. But I think that in Italy, you have one of the biggest metabolomics experts in the world, Vasilios Fanos, which does a lot of metabolomics.
Thank you.
So we have a question from Nicoletta De Simone. I've seen her hand.
Yes, yes. Hi. Thank you very much for your great presentation and your results. I just have a question. You showed that there is a difference in microbiota between control and gestational diabetes women in the first trimester of pregnancy. I'm wondering, I would like to know, what do you think about their condition before pregnancy? Do you think there is a difference even before pregnancy between physiological or normal pregnant patients, control, or women who develop during the second trimester of pregnancy gestational diabetes?
This is really, really a great question. I can answer part of it hypothetically because, you know, a lot of women that develop gestational diabetes start pregnancy with overweight. And we know that overweight women have a different microbiome than lean women. But what happens about the lean women that will develop gestational diabetes? And this is something that we are now trying to collect. And it's extremely hard to get samples from women prior to pregnancy and then follow them throughout pregnancy. And even after pregnancy, we have a study that is looking at postpartum depression. And, you know, getting these samples, following the women after pregnancy is really hard. So getting the samples before pregnancy is even harder.
Yes, I know that, but I think this can have more, of course, this information may have more data, more information about the metabolic and intestinal microbiota condition, even in terms of treatment.
I'm sure, I completely agree. I think that if we see such drastic changes in first trimester, it also happens before pregnancy. We can find it before pregnancy.
Okay, thank you very much. Congratulations for the presentation. It was great.
Thank you. Any other question? Professor Ferrazzi.
I have a question because sometimes we associate high cytokines with obesity and low-grade inflammation associated with obesity. What do you think about food inflammation in lean women?
About what? Sorry?
Inflammation caused by food. That is, inflammation caused, for instance, to exposure to lactose in lactase non-persistent women, or gliadins in women who are intolerant to gluten, or you know, this kind of conditions. Did you ever had a look at this?
We only looked at food allergies, and we published it in kids, okay, the kids that are allergic to foods, and it doesn't matter which food, whether it's milk, nuts, walnuts, eggs, sesame, they all lack a certain bacterium called Prevotella. And the interesting thing is, at the same time that we published this, there was a study published in Nature Communications, I think, that looked at pregnant women. And that study showed that women that have a lot of this bug, their children will not have food allergies. So I think, you know, that of course, food inflammation is mediated by the microbiome. How does this affect pregnancy complications? I have no idea, but it's a very good, you know, thing to check.
Thank you. Grazie. I think Gabriele was, we were discussing about, you know, microbiota, progesterone, and gestational diabetes. And one question that we were sharing was, what is happening, you know, after delivery in gestational diabetes? That soon after that, insulin resistance is back in place, and is overcome, and the glucose profile becomes normal soon after the placenta is removed and the progesterone production is reduced. So what do you think about what is happening after delivery in women with gestational diabetes?
So in women with gestational diabetes?
Yes. After delivery.
After delivery, yes. This is something we're looking at, and I still don't have the results, but I would assume that in 2 to 3 months, I'll be able to email you with the answers.
Okay. So the progesterone is a key player in changing the balance between firmicutes and bacteroides, also in these cases, is it?
And in the increase in bifidobacterium.
Yes.
Yeah. And again, we're now trying to understand how this is happening, you know, how does bifidobacterium sense progesterone? How has this evolved?
Okay. Okay, thank you. Gabriele?
I have another, not a comment, just a question about someone says that after almost 3 months later after cesarean section, the neonat has a complete restoration of the microbiota of how it should be. My question is, what is going to happen in this interval time, 3 months almost, in which the microbiota is not the original one, or what it should be established to be?
Good question, no answer. Nobody knows. Nobody knows. There are a lot of big groups studying this and trying to understand, you know, what I said, the long-lasting effect. For example, I showed you the study of infants getting antibiotics 2 days in the first 48 hours. Other studies which look at, for example, 3 antibiotics in 3 weeks of life show the exact opposite phenomena, shows that these babies gain more weight and become obese. So the critical, I guess, first 1, 2, 3 weeks are apparently, you know, life-changing. And I think in the coming 2-3 years, we'll see answers to your question. You know, what does this mean for sure, and not just correlative?
And excuse me, I have another question about a very simple issue, like a person like me that is always in the delivery room. I noticed the paper about the restoration of vaginal microbiome in babies coming from a cesarean section, but someone is going to say that the baby has a vaginal microbiome only if there is the complete the vaginal delivery. I mean, that the baby was passing throughout the vagina. What do you think about this hypothesis? Is it possible? Is just a theory and a hypothesis or something alike?
So we now, I think in the past year or 2 years, are understanding more and more that it's not the vaginal microbiome that is important for the inoculation. It's the gut microbiome. Mm-hmm. And the baby will get the important bacteria from his mom's rectum and not from the vagina.
Thank you. This is an extremely clear, step. I mean, it was said by Sant'Agostino. Sant'Agostino was said that nascimur inter feces et urinam. It means that we are going to be delivered. We are coming out between stool and urine. It was a very simple human condition, but probably is something that it was known or unconsciously known by the ancient.
Yes, so even wise, we now see evidence for that it's actually the gut microbiome that does the work.
Thank you.
I remember we were talking about the anthropological aspect of human delivery, and in nature, most of fetuses are delivered with intact membranes. Okay, so if we do not rupture the membrane at 8 centimeters, 87% of our newborns are delivered within an intact chorioamniotic sac. So my question is there a relationship between cutaneous microbiota and perineal anal microbiota? So that this explains why 2,000 years ago newborns were delivered with intact membranes, and the metagenome established at delivery was the cutaneous, perineal cutaneous, but not the vaginal microbiota. What do you think about it?
No idea. I really don't have enough data to answer that question.
It would be interesting to check the few cases who are delivered with intact membranes and see whether or not they are different. But well, I'm a bit older than you all, and I was delivered at home, okay? And I was delivered, we say you're a lucky guy because you're delivered with your shirt on, okay? So you're delivered with the membranes, okay? And in those times, that was a common way to say among, people, to say that you're a lucky guy because you were delivered with your shirt, with the intact membranes, okay? So that was common. It was not rare. So
I'll look into it, but if you're talking about birth at home, we definitely know that there are differences in the microbiome of infants born at home versus hospital, emergency C-section versus planned C-section. All of these give different microbiomes. And I think there's a paper coming out showing exactly that.
It would be interesting to sort out those few babies who are delivered within intact membranes and see what is happening to them.
I will go and talk to this author that I know that is going to publish and really look at it, but it's interesting, yeah.
Okay, Thank you. Gabriele, cosa dici? Ci fermiamo qua? Shall we stop here?
I think that we don't have other questions.
I think that it was, I think that Professor Coren was really exhaustive. I mean, the right definition of Professor Coren on this field is milestone. We have been really very, very lucky to have him with us, and it's always a pleasure. And today I learned something more about in his paper. It was really very kind of you to present your preliminary data on gestational diabetes. Thank you very much.
Thank you. And now we need to start collaborating.
Okay, I will talk to Fabio Mosca, the chief of neonatology, and I see some of his co-workers were listening to this webinar, and I will talk to him about the milk in the COVID vaccinated women, so that maybe they are interested in this. Okay.
Okay.
Thank
Great.
you. Grazie.
Thank you.
Thank you so much.
Thank you very much.
Bye.
Thank you. See you next Saturday with MJC with vaginal approach in hysterectomy. Thank you.
Grazie Agnese.